Hello, my crystals, and welcome back to my ASMR channel. I hope you are doing absolutely amazing and taking care of yourselves as best as you can. Today's video is going to be a little bit abnormal in that it's going to be a speed round of applying my makeup. I'm not late, but this was an impromptu and I'm just going to be doing a realistic makeup that I would go the speed that I'd be going if I weren't filming. So I'm going to start off spraying my face in a beauty blender. It's basically just water and doing my, what is this called? Halo Glow. So I am going to do some work today. If you're new here, hello, but I am in school for marine biology, and I have a part-time job working with coral, but today I'm going to be focusing on my own research. So I'm going into the microscopy lab, and I'll be using a transmission electron microscope to look at tissue within a sea squirt. I know, so random, so specific, but that is just one aspect of my project. So I want to try to find the bacteria that are living within the sea squirt. I want to try to visualize them using a microscope and the transmission electron microscope, also known as TEM, abbreviated. It helps look at very fine detail within organisms or tissue or any thin sectioned material. So I have dissected the sea squirt and fixed it with some preservatives, and I've already thin sectioned it. They are 90 nanometers thick sections, which I will then image on the TEM, the microscope. So all my samples are ready to go, and I did a trial run last week, and I don't really know what I'm looking at quite yet. So I did a little bit of background research on sea squirt ultrastructure and anatomy from previous literature to hopefully give me a reference of what I'm looking at so that I can identify the bacteria a little bit easier, if there are any microbes. I know I owe you a in-depth video on my research. This is just one small aspect of what I'm doing. I'm thinking I'll just run through my poster with you at some point, because that has most, if not all, of the information that I want to tell you about, since a lot of you seemed very interested in what I'm doing for my PhD. I just have not gotten around to it. reason. I'm doing my makeup way out of order today, but honestly, who cares? Who cares? So yeah, I'll be on the microscope for most of the day because it is kind of time consuming. The microscope is just, it functions differently than a regular light microscope because instead of light it uses electrons, so it takes a lot of energy, and it's a very precise instrument, so it needs various alignments done before it can even be functional. So it usually takes, I scheduled myself on it for 5 hours today, so I'll go in shortly and try to get some images and yeah, see what I can find. A lot of my PhD work is very exploratory and kind of venturing into the unknown. So it's pretty fun, but it's also can be frustrating because there's not a lot of previous work to reference or build off of. So I'm just kind of flying by the seat of my pants sometimes. But that is the beauty of science. You have to start somewhere, right? It's really exciting that I am able to spend my time doing this, just discovering the unknowns of sea animals and their microbial communities that live within them. I study symbiotic Archaea, which are a type of microorganism. They are one of the 3 domains of life. So you have your eukaryotes, which are like humans and animals and plants. And then you have another domain of life, which are bacteria, which are the microorganisms that you can't see that are living basically everywhere. And then in the 1970s, another domain of life was discovered. These are also microorganisms that look like bacteria but are in no way related to bacteria, so they are their own branch of life. And interestingly, they are more closely related to eukaryotic organisms like humans, animals, plants. So they're very cool. And being discovered in the 1970s, that's pretty recent for science. So they were just discovered 50 something years ago. So I study a specific type of Archaea that are very dominant in soils and in marine environments. So they're very abundant in the ocean, and they have been discovered in marine sponge organisms, and quite recently, within the past 10 years, a study or a few studies excuse me, have found that they are also present in sea squirts. So sea squirts are marine invertebrates. They are also referred to as tunicates or ascidians, and they are the organism that I'm focusing on when I'm looking at my specific type of Archaea. So the specific type of Archaea that I look at convert ammonia, which is a nitrogenous compound, into nitrite, which is a different nitrogenous compound, but it's basically recycling ammonia from the water and converting it into nitrite. So it's kind of like a waste removal organism, and they are very beneficial to marine organisms that cannot survive in high ammonia level concentrations in the ocean, because high ammonia can be toxic to fish and other wildlife. So they are a very good microorganism. They are in no way, shape, or form bad or pathogen. Funny enough, no Archaea have been found to be pathogens. So when you think of bacteria, you know, you have your good bacteria and your bad bacteria that can cause disease. But with Archaea, there are no disease-causing organisms that have been discovered thus far. So these are very beneficial, good microorganisms, and I'm so happy I get to study them because not a lot is known about them still. Okay, so I think this is going to be the completed look, and I just did a little ponytail braid and everything. But thank you so much for listening to me ramble. I will do a very in-depth video on my research shortly. I know I was rambling, but that just skimmed the surface of everything that I want to talk about. So I will see you very shortly in my next video. Remember that you are loved and abundant always. Always, 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 always. I will see you in my next one. Good night.